Hello, I'm your host, Jackie Mullins, and welcome to From Cells to Us. How? Last episode, we finished the full city tour. Nucleus, mitochondria, ER, Golgi, lysosomes, cytoskeleton, motor proteins, the whole infrastructure. We have a fully equipped eukaryotic cell ready to do something no prokaryote ever managed: work with other cells. But before we get there, we need to talk about something that happened first, something that changed how genetic information gets passed on, something that unlocked a level of diversity that made complex multicellular life not just possible, but kind of inevitable: sexual reproduction. But remember, we are still roughly a billion years ago. We are still single-celled organisms. There are no animals, no plants, there are certainly no humans, and yet sexual reproduction evolved here, which raises the question: why? Why would evolution invent sexual reproduction? Why would any organism give up the most efficient reproductive strategy that exists, just copying yourself, in favor of something far more complicated, far more expensive, and far more risky? Well, that's what we're going to discuss today. Let's get into it. So before we can understand why sexual reproduction evolved, we need to understand what it was up against. Because cloning yourself, asexual reproduction, is genuinely a great strategy. And the proof is in the pudding, so to speak, because asexual reproducing organisms started this whole life thing, and they are still kicking it today and giving us one heck of a showdown every winter. And asexual reproduction is a smart way to go about things. Just think about it. You are an organism that survived. You have figured out how to eat, avoid being eaten, and reproduce in your environment. Your genes work. Your body works. Why would you mix those winning genes with someone else's? Why would you take a perfectly good instruction manual and scramble half of it with a stranger's instruction manual and hope the result is better? And I feel like we all know that one person, you know, the type who, if given the chance, would be like, yep, nothing better to add here. My children should be 100% me. Because here's the math. Every time you reproduce sexually, you are only passing 50% of your genes. Clone yourself and you pass 100%. And maybe you're picturing some giant wuss of a bacterium, you know, and thinking, well, this little fella probably shouldn't be passing 100% of their genes. They should definitely mix with someone better. But that's not how selection works. That wuss of a bacterium will just die. Selection is not going to be like, but one time it beat 10 opponents in bacterium chess, or throws really well in bacterium football. No, selection is cutthroat. You're good at surviving or you're gone. In the Game of Thrones, you win or you die. So from a pure numbers standpoint, cloning wins every time. You are literally twice as efficient at spreading your genes if you just copy yourself. Plus, there's no added time of finding a partner. Now, you might be thinking, okay, Jackie, like I know what you're doing, but sexual reproduction is obviously the choice for us. I know this because we're multicellular. We reproduce slowly, we have small numbers of offspring, we live long, complicated lives. Of course, sexual reproduction makes sense for us. We need the variation. Bacteria can afford to play the lottery because they buy 1 billion tickets at a time. We only get a handful. And when you only get a handful, you better make sure each one is as genetically interesting as possible. And you're right. For complex multicellular life, once you understand variation and natural selection, the advantages of sex aren't that hard to see. We're too complicated and too slow to rely on random mutation alone. Recombination does the work instead, generating variation on purpose every generation, without waiting around for lightning to strike. But here's the thing, that's the easy question, because sexual reproduction didn't evolve in multicellular organisms. It evolved long before any of that existed. It evolved in single-celled eukaryotes, and that is where the real mystery is, because in a single-celled world, where organisms are smaller, faster, and reproducing more frequently, the case for cloning is even stronger. So why did sexual reproduction show up at all? And once it showed up, why did natural selection keep it? That is the question this episode is trying to answer today. But before we get into the mystery, let's make sure we understand what sexual reproduction actually does at the biological level. Sexual reproduction isn't purely about reproduction. I mean, well, it is. That's the whole point. Reproduction. Now. But we're not talking about now. We're talking about a single cell a billion years ago. And back then, sexual reproduction was all about genetic recombination. And genetic recombination is the shuffling of genetic material between two individuals. That's it. Meaning there is no official offspring. So with sexual reproduction in the single cell world, they didn't produce another cell, right? Like what? Reproduction is what happens afterwards. The actual innovation is the shuffling part. So sex didn't evolve to make babies. It evolved to fix problems. Babies were just kind of like the side effect. But wait, you might be thinking, if two cells merge and divide back out as reshuffled versions of themselves, how did the population actually grow? Like if every time two cells got together, they just became two different cells, you'd never get more. You'd just keep trading partners forever. And the answer is sex was never the population growth strategy. Asexual reproduction was still running the whole time. So sex was like the side dish. It wasn't the main course at this point. Most of the time, these single-celled eukaryotes were just dividing normally, cloning themselves, growing the population, doing the boring, reliable thing. But periodically, under the right conditions, stress, parasites, something in the environment turning hostile, they would trigger into a sexual cycle. Two cells merge, recombine, divide back out as new shuffled versions, and then go right back to cloning. So asexual reproduction was still the population growth engine. Sex was the variation engine. Two completely separate tools running the same organism for completely different purposes. So they only trigger the sexual cycle when things get hard. It's almost like sex was the emergency protocol, right? Break glass when cloning stops working. So sex wasn't replacing asexual reproduction. It was just an occasional addition to it. The question isn't why did they give up cloning for sex? It's why did they bother adding this expensive extra step at all? And that is what we're trying to figure out. And what it basically boils down to is in evolution, when you can't outrun your enemies, you outsmart them by never looking the same twice. So how does this actually work, though? I'm sure some of you have an idea on how multicellular reproduction works, but how does this happen with single cells? There's no sperm, no egg, no pregnancy, so how? Well, what happens is two single-celled organisms simply find each other, touch, and merge. So are they seeking each other out? Do they have their Match.com profiles? Are they just hoping that the single cell of their dreams will answer their call? And the answer is kind of. You were expecting me to say no, weren't you? But some single-celled eukaryotes, yeast being the best example, actually release chemical signals when they're ready to combine. Pheromones. They're broadcasting. And this shouldn't be such a stretch to imagine because animals do this, right? Mating seasons and whatnot. Once they release their signals, nearby compatible cells detect those signals and move toward the source. It's pure chemistry. One cell releasing a signal, another cell's membrane detecting it, and the fusion process initiates automatically. Saying we have chemistry in a relationship takes on a whole new meaning now, doesn't it? Now, I said a minute ago that compatible cells detect these signals. The Match.com signals that they're sending out. And that word compatible is doing a lot of work here because not just any two cells can merge. Even 1.2 billion years ago, these single-celled organisms already had something like mating types. It wasn't male and female exactly, but different chemical structures on their surfaces determined whether fusion could happen at all. The machinery for finding the right partner was there from the very beginning, which is pretty wild to think about. So again, how it was decided if two cells could mate or not is membrane compatibility. Some can mesh, some can't. That is the deep ancestral root of biological sex as we know it today. Male and female, sperm and egg. All of that elaborate machinery, all of that complexity, all of it traces back to this moment. 2 membrane types that could mesh and 2 that couldn't. That's it. That's where it started. Bam, you just learned the origin roots of male and female. Who knew it happened before we even crawled out of the ocean? But why? Why did there have to be compatibility at all? Like, so what if 2 star-crossed lovers wanted to be together? Who are we to judge? But the answer is actually built into the problem sexual reproduction was trying to solve in the first place. If any 2 cells could fuse indiscriminately, you'd immediately run into 2 problems. 1, cells that are too similar, basically clones of each other, don't gain anything from fusing. You shuffle 2 identical decks and you just get the same deck. There's no new variation, no benefit, pointless cost. That's just asexual reproduction with extra steps. If you need physical evidence, just look at King Charles II of Spain or Joffrey from Game of Thrones. We are hot on Game of Thrones today. And second, cells that are too different from completely unrelated lineages create the opposite problem, which is that they just don't work. It's like putting a gaming token into a vending machine and wondering why nothing's clicking. Now, I know some of you smarties might be thinking, wait, Jackie, didn't you say all cells speak the same genetic language? And yes, they do. The DNA code is universal, but speaking the same language doesn't mean you're running the same operating system. Think about it like Mac and Windows. They both speak binary code underneath. That's your universal genetic language. But try running Mac software on a Windows machine and see what happens. The underlying language isn't the problem. The operating system built on top of it is. In cells, the operating system is the regulatory sequences, the DNA that tells genes when to turn on and off. And in cells that are too different, those instructions conflict with each other. It's like 2 toddlers. One wants the light on, one wants it off. They keep switching back and forth until neither one remembers which one wanted it on and which one wanted it off. The cell can't coordinate itself and it just falls apart. And on top of that, wildly different organisms have a different number of chromosomes. And in meiosis, we'll get into that part in just a bit. In meiosis, it requires chromosomes to pair up and find their partner. If the numbers don't match up, the shuffle breaks down completely. Think of that like the poor kid at the school dance with no partner. Except instead of just standing there sadly, in the cell, everything just falls apart. Because everything needs a pair. No chromosome left behind. So what you need is compatible but not identical. Different enough to generate useful variation, similar enough that the combined genome can actually function. So kind of like that person who's trying to get over someone and they pick someone kind of close and everyone sees it but them. Like in Friends, when Rachel went out with Russ, who was essentially a Ross doppelganger. Similar enough that the chemistry was there, but not so identical that it was pointless. That's your sweet spot. Not twins, not strangers from different planets, just different enough to make things new. And here's something that might be rattling around in your head right now. Wait, Jackie, didn't you say bacteria do horizontal gene transfer across wildly different organisms, like grabbing DNA from completely unrelated cells, picking antibiotic resistance from a completely different bacteria? You know how we joked that from a stranger you could get the trait of how to play the piano on the street or something? How does that work if incompatible operating systems are such a problem? And that is a great question. Horizontal gene transfer works across wildly different organisms precisely because the universal genetic language is enough. You're not merging 2 whole genomes, you're just inserting a snippet. The receiving cell's machinery reads it in the universal code, recognizes it as a valid instruction, and runs it. No operating systems clash, no chromosome pairing problem, just a small addition to an already functioning system. But sexual reproduction is a much bigger ask. You're not inserting a snippet, you're taking two entire genomes and mixing them up, then shooting them out and expect them to create life again. The universal language is just the floor, the minimum requirement. Horizontal gene transfer walks into the building, orders a martini because that's the HGT style, and chills in the lobby. Sexual reproduction needs to get on the elevator and clear hundreds, sometimes thousands of floors all the way up to the top. Same language, completely different level of commitment, which is why horizontal gene transfer crosses the big boundaries. Every cell has that first floor lobby. They can order their martini and do what they came to do without anyone checking to see if they have a visitor's pass. Sexual reproduction, on the other hand, has to have a special badge for each floor, making sure each is within compatible operating systems within organisms similar enough that the merge could actually work. Or think of it like this. If you ever wanted to cheat on a paper, and I know none of you have, nor have I, but you wouldn't take an entire paper from the internet and try to mesh it with your whole paper. One, it wouldn't make sense. And two, you'd definitely get caught. But if you went through that other paper and carefully inserted snippets into yours, well, now it flows. Now you won't get caught. And your paper just got a whole lot better. That's horizontal gene transfer, small insertions into an already functioning document. Sexual reproduction is trying to merge two entire papers into one, which only works if they're already pretty similar to begin with. And we can actually see this playing out in the world today, because that compatible but not identical sweet spot, the thing that those ancient single-celled eukaryotes were navigating, is exactly why different species can't reproduce together. Or if they can, why those offspring are sterile. Think about the horse and the donkey. They're similar enough to produce a mule. The operating systems are close enough that a functioning organism can be built. But the mule is sterile. But why? Is nature just like, I shall give ye this one, but thou mule shall hold no lands, nor bore no children? No, here's what actually goes wrong mechanically, because it's not dramatic. The mule doesn't try to reproduce and explode, right? It's much quieter than that. When reproduction occurs, the horse contributes 32 chromosomes and the donkey contributes 31. The chromosomes simply cannot find a proper partner. And we'll get into exactly how that pairing works in a minute, but the short version is 63 chromosomes can build a body just fine. However, what is produced just can't make a new one. Thou surname ends with this mule. Withist is probably not a word, but it is today. All right, and then there's the even further apart scenario, like a cat and a dog, a human and a fish. These don't even get to the pairing problem because they don't even get to that part. The failure happens so much earlier at fertilization or right after. Even if you somehow got the genetic material from two completely different species into the same cell, the molecular machinery that reads the genome and starts building an organism would immediately run into chaos. Those regulatory sequences, the parts of the genome that tells the genes when to turn on and off, the toddlers playing with the light switch, in a cat and dog combination, those signals are so different that the most basic instructions conflict with each other. Turn this gene on. No, turn it off. Build this protein now. No, don't. The incredibly precise choreography of signals that tells a fertilized cell how to start dividing and differentiating and becoming something falls apart almost immediately. There's no mule equivalent here. You're not going to get a cog or a dag, which could either be a dog and a cat mix or just the dog from Snatch if Brad Pitt's character is saying it. The mule at least got built. The cat and dog combination never even breaks ground. So if anyone has seen that Nickelodeon show Cat Dog when you were younger, I am sorry to be the bearer of bad news, but it's completely fabricated. Not scientifically based at all, surprisingly. So the reason species boundaries exist reproductively traces all the way back to what those ancient single-celled eukaryotes were doing 1.2 billion years ago. Compatible, but not identical, is the sweet spot. Too similar and recombination is pointless. Too different and the system breaks. All of it tracing back to two single-celled organisms in the ancient ocean where membrane proteins happen to recognize each other. It just didn't have eyes yet, or a face. Okay, well, let's get into the mechanics, though. I'm sure some of you were not satisfied with my whole we throw some DNA in a bucket and then it mixes and gets taken out. So how does this actually work? Well, today we call the process that makes sex cells meiosis. And we all know the end result. I don't have 3 mini Jackies running around. I have 3 very different children who kind of look like me, who kind of look like their dad. 1 is immune to strep, like me. 2 aren't. All 3 have my eczema, all stemming from my mom. Luck of the draw. But why? How does that happen? And why do we need meiosis? Why can't we just smush 2 skin cells together and make a baby? Well, here's the thing about your skin cells, your liver cells, your brain cells. Every single one of them contains your complete genome. All of it. Both copies of every chromosome. It's like these cells are carrying around a full manuscript of a story. 46 pages long, we'll say, as you have 46 chromosomes total. 23 from your mom, 23 from your dad. Now, maybe you think your parents skimped you. Why didn't you give me all of your genome, ma and pa? Well, if they did, say it was possible to use a normal cell with all 46 chromosomes to make a baby, like a skin cell. The baby would start with 46 chromosomes, then add the other person's skin cell, another 46. Now you've got 92. Their kids would have 184. Within a few generations, you'd have organisms with thousands of chromosomes and the whole system would collapse under the weight of 1 billion chromosomes. Now there are some other issues there too, but there's somewhere to start. So before reproduction can happen, something has to cut the number in half. That something is meiosis. Meiosis is a special kind of cell division that happens only in the reproductive organs, only for the purpose of making sex cells, sperm and egg. And it does 2 things that regular cell division never does. First, it cuts the chromosome count in half. Your 46 chromosomes become 23, half a genome. Half the manuscript at only 23 pages. An incomplete set deliberately designed to be completed by someone else's half. 'Tis very romantic, no? Second, before it cuts the count in half, the chromosomes do something remarkable. They basically put on some country music, and they have the 23 pairs line up and do-si-do. And in the do-si-do, when the chromosomes come together, they swap bits of DNA all along their length. Not just one swap, multiple swaps, all the way down. So if you came in wearing all pink and your partner came in wearing all blue, by the time you separate, neither one of you is fully pink or fully blue anymore. You're striped, patchy, a combination that never existed before in exactly that pattern. And then remember the cytoskeleton from last episode. Those microtubules. Well, spindle fibers of microtubules show up and absolutely destroy this fun dancing scene. Like someone coming in at the end of a show with those hooked canes from Looney Tunes, and just pulling them off the stage. They grab each X by the middle and pull. The pairs get ripped apart into separate cells. And then those cells divide again. So you started with 1 cell and your full genome. And you end up with 4 cells, each carrying only half, 23 chromosomes instead of 46. They only have half the manuscript, but not the original half of the manuscript you inherited from your mom and dad. It's a new one, a one looking for a different ending. Those are your sex cells. That is what meiosis made. And that is why my kid got eczema and not strep immunity. So to sum up, you are born from an egg and a sperm, each carrying 23 chromosomes, a unique pairing and mix-up of your grandparents, 2 from each respectively. They combined and form you. Now you have 2 chromosomes from that mix-up, but you are not a mule. You can reproduce. Thou surname shall continue. So your body starts taking cells with full chromosomes and turning them into cells with half chromosomes so that when the time comes, you can create a new being. That is what meiosis does. It's not just cell division. It's a deliberate, elaborate machinery for generating novelty every single time. in contrast to asexual reproduction, which trust speed and numbers to give them genetic variation. So how did single-celled eukaryotes do it? Well, same basic idea, way less drama. Two cells find each other, fuse, and briefly become one cell. Two copies of everything jumbled together. But remember, sexual reproduction is basically an emergency situation. So they're in and they're out. Break glass, do the shuffle, get out. So almost immediately, the chromosomes pair up, swap chunks, and divide back out into the world as new reshuffled single cells. And there's not attachment, right? No additional body built, no pregnancy, no childhood. Two cells went in, the shuffle happened, new reshuffled cells came out. Done. Same crossing over, same novelty, just faster, simpler, and without all the elaborate machinery we built around it later. That came when multicellular life showed up and needed meiosis to do a whole lot more than just shuffle and bolt. Think of it like two strangers meeting during a crisis, quickly trading something useful and then walking away in opposite directions. There's no relationship, no follow-up text, just, here's some of my stuff, I'll take some of yours. Good luck out there, see you never. So back to the question on why single-celled organisms would ever start mixing its genetic material with another organism, paying the cost to do it. Scientists have been wrestling with this question for decades, and there is no single clean answer. The honest answer is probably several pressures operating at the same time. Here are three of the leading hypotheses. One is called the Red Queen. The name comes from Lewis Carroll's Through the Looking Glass, where the Red Queen tells Alice that in her world, you have to keep running just to stay in the same place. And that's exactly what's happening in evolution. Organisms are not evolving in isolation. They are evolving in response to each other. Every time one evolves a new trick, the other evolves a counter, an arms race with no finish line. If you stop running, if you stop evolving a counter, you're eliminated. You are the weakest link. Goodbye. And parasites, bacteria, virus, fungi, evolve extremely fast. Their generation times are measured in hours or days. They can evolve new ways to attack a host in the time it takes that host to produce a single offspring. Now imagine if all our offspring were genetically identical. Any pandemic, any single pathogen that figured out how to beat one of us would beat all of us. If you had 10 kids and one caught something fatal, you just have to gather the family and be like, well, Tiny Tim didn't make it, and since we're all genetically identical, I'm so sorry. I should have diversified. Goodbye, everyone. We're here because that didn't happen, because the deck got shuffled. The host population of identical clones is a disaster waiting to happen. A parasite that figures out how to breach one clone's defenses can breach all of them. The whole population goes down. But a population with genetic diversity, the parasite that cracks one individual's defenses might be stopped cold by the next individual's slightly different configuration. The shuffled deck keeps the parasite guessing, keeps it from finding the master key. And here's the critical thing. This pressure was just as intense in the single-celled world as anywhere else. 1 billion years ago, the oceans were teeming with viruses and parasites evolving at a blistering speed. A population of single-celled eukaryotes that cloned each other was just as vulnerable to being wiped out by a single pathogen as any population of clones would be. Just because Eugene the eukaryote wasn't multicellular didn't mean he wasn't fighting for his life. Didn't mean he wouldn't need something to fend off these parasites. The arms race was already running hard. So, in the Red Queen hypothesis, it states that sexual reproduction is essentially an immune strategy, a way of staying one step ahead of the things trying to kill you by constantly regenerating new combinations that parasites haven't seen before. The next hypothesis is DNA repair. And this hypothesis states that the earliest function of sex wasn't variation at all. It was actually fixing broken DNA. DNA gets damaged constantly. Radiation, chemical exposure, copying errors. And if you're asexual, when a critical gene gets damaged badly enough, you're just stuck with it. You pass that damage on. But when you combine genetic material from two individuals, you get a second copy to work from. Broken gene on one strand, no problem. Use the other template, fix it, move on. So the DNA repair hypothesis states sexual reproduction was first used as a repair kit, shuffling engine second. The variation may have almost been a side effect of solving a much more immediate problem, keeping the genome intact. Then there's the third hypothesis, and that's called Muller's ratchet, named after geneticist Hermann Muller. Here's the idea. Without recombination, harmful mutations accumulate over generations with no way to purge them. Each generation is slightly worse than the last, like a ratchet clicking one notch forward with no way to click back. Small errors pile up, the genome slowly degrades, sexual reproduction stops the ratchet. When you combine genetic material from two individuals, you can produce offspring that have shed some of the accumulated bad mutations. So it's not just about adding good things, it's about being able to get rid of the bad ones. So basically what that's saying is that when the chromosomes do the meiosis do-si-do, the bad mutations only go to some of the cells, not all four. So you have less of a chance in getting a bad mutation. For fast reproducing bacterium with a tiny genome, this is more manageable. But eukaryotic cells have vastly larger, more complex genomes. More DNA means more places for errors to accumulate. The ratchet problem gets worse the more complex you get. And sex was already solving it before complexity really took off. And here's the wild part. We don't have to theorize about this. We can actually watch it happening in real time. In your kitchen, in your bread, in your beer. Yeast. Yeast are single-celled eukaryotes, and they do exactly what we think those ancient single-celled eukaryotes were doing 1.2 billion years ago. Most of the time, these yeast cells reproduce asexually, just budding off in little copies of themselves, cloning away happily, passing on 100% of their genes, living their best life. But when things get hard, just picturing a poor little yeast bud on the streets, hungry like Oliver Twist, or during the plague, that's when they break glass for an emergency. So when they're starving or the environment turns hostile, or something is threatening the population, they switch. Two yeast cells will fuse together, combine their genetic material, shuffle the deck, and divide back out as two new genetically reshuffled cells. They don't do it because it's more efficient. They do it specifically when efficiency isn't saving them anymore. This is sex with no babies, pure recombination, exactly what the original innovation actually was before multicellular life tangled it up with reproduction. These organisms are living fossils of the original invention. They're showing us what sex looked like before it became what we recognize today. And crucially, they turn it on under stress. When parasites are winning, when the environment shifts, when the clonal strategy stops working, which is exactly what you'd predict if the Red Queen, DNA repair, and Mueller's ratchet were the real reason sex got selected for in the first place. I mean, this is the equivalent of Inigo Montoya saying, "Because I know something you don't know, I am not left-handed," and then switching to his right hand and dominating. That's what sexual selection did for these organisms. The evidence didn't stay buried in fossil record. It's been sitting in your sourdough starter, the whole time. Okay, so here is where we land. Cloning is more efficient on paper, and in a world of fast reproducing enormous population organisms like bacteria, cloning works. They reproduce fast enough that random mutations generate variation anyway. They have horizontal gene transfer as an additional workaround. Cloning got patched into something viable through sheer scale and speed. But single-celled eukaryotes were already different. Bigger genomes, more complex machinery. You can't treat a studio apartment and a mansion the same way. Like if you hired someone to clean and tried to charge them the same amount for a mansion and a studio apartment, they'd be like, "No, are you crazy? That's not happening." Eukaryotes are slower than bacteria, more vulnerable to accumulating errors, and living in an ocean absolutely full of parasites evolving at speeds they couldn't match through random mutation alone. And in this treacherous environment, 3 problems landed all at once. Parasites, running a faster arms race than random mutation could keep up with. Genomes, getting damaged with no backup copy to repair from. Mutations, accumulating with no mechanism to purge them. Sex solved all 3 simultaneously, and it did it consistently every single generation, built right into the reproductive process. The cost is real, but the alternative was a genome slowly degrading while parasites evolved 1,000 times faster. Sex wasn't just worth the cost. In the eukaryotic world, it was the only viable long-term strategy. All right, and once sex was established in single-celled eukaryotes, it sets up everything that came next. A sexually reproducing population generates massive variation every single generation, unlike clones who have to sit around waiting for a useful mutation to randomly show up. It's like being hungry and knowing your partner already ordered food. You don't know exactly what's coming, but something is definitely on its way. Versus just sitting there and staring at the door hoping DoorDash shows up by accident. But sexually reproducing populations generates massive variation every single generation. Natural selection suddenly has an enormous amount of raw material to work with. Useful combinations appear constantly. Harmful ones get weeded out. The whole process accelerates. And what comes next in our story requires exactly that acceleration. Multicellularity. Organisms made not of one cell, but of trillions of specialized cells working in coordination. Eyes, nervous systems, immune systems, brains, none of it is buildable through random mutation alone at the pace complex organisms reproduce. You need a system that generates variation on purpose, reliability every generation. Sex was already doing that. It had been doing it for hundreds of millions of years before the first multicellular organism appeared. Sex didn't cause multicellularity, but it built the engine that made multicellularity possible. Now, and here's something I wanted to clear up. That engine, that ancient single-celled logic to clone most of the time and shuffle when it matters, it's actually still running inside of you right now. Your liver cells are copying themselves. Your skin cells are copying themselves. Your immune cells are copying themselves. Every time a cell in your body divides to grow, to heal, to replace old cells, it copies its entire genome and splits into 2 identical cells. 1 becomes 2. Same DNA, perfect copy. That's cloning. That's asexual reproduction happening inside your body billions of times a day. We call it mitosis to make it sound fancier. So you are not purely a sexually reproducing organism. You are mostly an asexually reproducing organism who reserves the sexual reproduction, the shuffling, the meiosis, the novelty generation, for one very specific job: making the cells that carry your genes to the next generation. Sperm and eggs, that's it. Everything else is still just copying, which means those single-celled eukaryotes we spent this whole episode talking about, cloning most of the time, triggering the sexual cycle only under pressure, weren't doing anything fundamentally different from what your body does today. The logic never changed. It just got more elaborate, more specialized, more you. So let's bring it all the way back. 1 billion years ago, in the ancient ocean, a single-celled eukaryote did something no other organism had done before. It combined its genetic material with another organism's. Not because it was more efficient, not because it made obvious sense on paper, probably because parasites were picking apart every clonal population that couldn't shuffle the deck. Because genomes were degrading without a second copy to repair from. Because mutations were accumulating with no way out. It was expensive, it was complicated, it cut gene passing efficiency in half, and it changed everything. The best defense against a rapidly evolving enemy is to never look the same twice. The Red Queen keeps running, the deck keeps shuffling, the combinations keep coming. You are the product of over 1 billion years of shuffling. Every ancestor you have ever had, going all the way back to the first single-celled eukaryote who in the ancient ocean managed to find a partner, combined their genetic material, and produce offspring that survived long enough to do it again. The odds of you specifically existing are essentially zero. And yet here you are. Next episode, we are finally leaving the single-celled world behind. After 11 episodes of building the machinery, of perfecting the systems, evolving the strategies, it's time for the cells to start working together. Multicellularity, the origin of bodies, how one cell became many. That's episode 12. I'm excited. So, some quick notes before you click off. One, thank you again for everyone who listens. And a small favor, if you're enjoying the show, would you mind following, rating, or the big one, even dropping a comment? Those would, one, make my day, and two, when you do any of those, the algorithm sends to new people. So by doing any or all of those, you're helping me to grow my show, and I would very much appreciate it. Second, I'm officially moving new episode days to every other Wednesday. It just works better with my schedule, except today, obviously, which this one is out on Friday. So I will be getting back to the every other Wednesday, not next week, but the week after. So if you were expecting Mondays, I apologize. It will be every other Wednesday from here on out. And last, if you want a visual to go along with the auditory experience, I have an Instagram and a YouTube channel where I make videos from some of the episodes. Links are in the description. You can check them out when you want. And thank you so much for joining me on this biological journey. I'm Jackie Mullins, and this has been From Cells to Us. How? I'll see you next time.